contact me, email me, whatever the case. Um, at this point, I'm going to keep all the due dates the same in terms of the paper. I remember, don't forget about that paper. Get working on that. Everything, luckily in this class, everything can be pretty much online, so exams shouldn't be affected, any of that kind of stuff. But once again, if you have any questions, let me know. So let me go over some of the stuff in the learning chapter. The This chapter deals basically with how the brain can change due to different types of experience. Okay, so learning um, psychologists define learning as a change in behavior, a relatively permanent change in behavior due to experience. So the idea is that we know at a behavioral level, like with classical conditioning, operant conditioning, and stuff like that, and other types of learning, that behavioral responses can change over time based on the experiences that you have. But what's sort of the mechanism of that? Is there a physiological basis for, let's say, classical conditioning or changes that occur in operant conditioning and stuff like that? And that's kind of what, at least partially, what your text is going into. And also, this notion of plasticity of the brain. I don't know if I said this in class before, but the basically the brain is not fixed. In terms of its hardware, so to speak. It's not fixed. It's not like a computer that you got that silicon base that it's all sort of fixed. And in order to upgrade your computer, for the most part, you almost have to buy another device. Humans, and other physiological, and mammals, primates, whatever, we can, as we learn, the brain can change. Now, I loaded up some audio file on the website of HM, and you can see here HM's anterograde amnesia. This is something I want you to listen to that audio file because you hear from HM, who was a person who, as you can see, suffered from severe, terrible epilepsy. Now, nowadays, epilepsy is treated primarily through, drugs that more or less reduce brain activity, all right, like things like gabapentin and stuff like that. But back in the 1940s and 1950s, when HM was younger, they didn't have that kind of stuff, so they used surgery. So what they found was, okay, we'll take out these temporal lobes, all right, because that's where the seizuring was. So he got a bilateral medial temporal lobectomy, all right, so it's essentially removal of big chunks of the brain, in particular the hippocampus and the amygdala. Okay, the hippocampus was explored in this chapter. The hippocampus basically is involved with getting what we call consolidation, getting memories, the experiences that you're having right now, get transferred into a long-term storage. And also it removed part of his amygdala, which I probably mentioned in class. This is a structure that plays a role in things like aggression and fear. But at any rate, his convulsions did drop, so his epileptic seizuring dropped, but he had profound amnesia. And you can see there's a picture here of a medial temporal lobectomy. So here you see this is the temporal lobes right here, and they kind of wrap under temporal lobes, and they took out a big chunk of his brain. Now, this is, I think, the portions of the medial temporal lobes removed from HM's brain are illustrated here, and that would be the pink areas. So when these areas were taken out, he had a really hard time remembering anything new. I mean, and it was a profound loss. Once again, they go into that in the audio file that I've uploaded on the class, and I think it's under the do first activity. So I want you to listen to that. And here you can see your author's going into some different types of tasks that we can use to test different types of memory. So if you take a cognitive psych class, you'll learn very quickly that there are a lot of different kinds of memory. And HM was, he had profound declarative memory loss. So he couldn't tell you what he just had for lunch that day. Let's just say if it's been like a couple hours or whatever. His whole experience was just sort of the here and now. He could not remember on a more of a declarative conscious level anything. But one of the things that was kind of, so the reason why I'm talking about this is because the hippocampus, we learned a lot about the hippocampus based on HM's case study. Here you can see, though, there's other types of memory, though. There's like muscle memory or memory for procedures. And here you can see the mirror drawing task. And you can see this is kind of tough to do. You put a mirror up in front, you got to draw looking at the mirror. HM would be given this. And what they're showing you here is that he improved in the task day by day, but he still never remembered actually doing the task. So there is memory here. It's more of an unconscious, almost like motor memory. It's kind of like if you imagine learning how to ride a bike, and you get better every day or every week, but never remembering that you ever rode the bike. That's kind of what's going on here. And you can see there's some. I'm not going to go through all of this here, but these are just some more ways to assess brain function. But here you can see some of what is important about this. The medial temporal lobes are very important in memory. We have different, and it's important for this consolidation process. Consolidation is getting stuff from a short-term storage to a long-term storage. So that's one type of memory. All right, and so when you've had, he had this profound amnesia. And, once again, your book, I want you to go through, you can see the different kinds of memories. Here you can see episodic, semantic. Episodic memory is what's really hit with the temporal lobe amnesia. This one, once again, it's just remembering what the events, the episodes of your life. You just couldn't remember that kind of stuff. Semantic deals more with the understanding the meaning of an event. That's a little different. Here you can see some other cases. RB is another one. This person had some sort of stroke, and it caused some. And after death, they could see what happened to this person. And because you can have memory loss with a stroke, or an inability to form new memories with a stroke if the temporal lobe gets damaged in the stroke. And here you can see some other disorders that are mentioned in the text. Here's Korsakov's. It happens to some folks who drank a lot. You get a thiamine deficiency, and they get brain damage essentially. So you can look at that. Alzheimer's, yes. Alzheimer's gets a lot of funding. A lot of research has been poured into Alzheimer's disease. We know more about it than we did 20, 30 years ago. Still no cure, but getting there. Every time I ask students if they know folks with Alzheimer's, a lot do. It's a relatively common problem if you get past 80. Hopefully you don't get it, but it's just relatively common. And here you can see, initially, you get these little tiny lapses in memory. Maybe, I don't want you to think that because you forgot where you put your car keys that you have Alzheimer's. That's not necessarily true, but you start to see this progressive loss of memory. But there's all kinds of physiological changes that occur. You get degeneration in parts of the brain. You get reduced acetylcholine output. Now, acetylcholine is important for attention and memory function. And as a matter of fact, one of the ways to make the symptoms better is to boost acetylcholine production. At least that's one way to do it. And so, your text goes into this a bit more. Maybe some of you, usually I get one person wanting to do an Alzheimer's paper. There's a lot out there about that disorder, but it's basically like a very, very severe form of dementia. And it's also got very specific types of anatomical changes. But basically, you're getting less brain, essentially. You're losing. Degeneration means that you're getting a loss of cells in certain parts of the brain. It eventually affects the whole thing. Here you can see post-traumatic amnesia. This is a head injury if a head injury or a concussion can cause this, could cause a brief amnesia, things like that. Here you can see concussion-induced amnesia. This happens, you can see the boxing, blow to the head, what they call it, getting punch drunk or whatever. When people are getting hit in the head, it's obviously going to potentially cause damage. And you can see through all that. Now this is definitely important, consolidation. Older memories stored in a more permanent form, okay? So once again, getting from that short term to the long term. Hippocampus seems to be important, okay? The idea is that the hippocampus is going to help process that information, those experiences, so that it can eventually be stored in the long term. And this concept here is the engram. This is the notion that there is a physical representation in the brain of your experiences, of your memories. That's the engram. Change in the brain that stores memory. So the search for the engram really has been focused a lot on the hippocampus and some other parts of the brain. So that's it. Literally, this search has been going on, for a long time. I mean, this has been 40s and 50s. Neuroscientists were interested in this. Nowadays, with the optogenetics, that stuff has really provided provided more evidence for this engram, which I'll get to. Okay, let's just see. I don't want to get into. I don't want to go through all of this stuff. I want to just hit some of the major points here. You're seeing different. How can we test memory function or learning function? With humans, you saw some of the things like with the beginning of the chapter there, but with monkeys, you can do what's called a matching to sample and stuff like that. Monkey is presented with some sort of object that's unique. Underneath that is food. You got to remember, animals work for food. That's what they want. That's the reinforcement. This is you're forcing the animal to remember a little bit in order to get the answer right. That's basically what's going on there. If they get it correct, if they can remember it, they get it correct, they're going to get food. All right, so that's one way to so a neuroscientist might train an animal with this task, and once they're good at it, then they can go in and possibly damage part of the hippocampus. And what you'd find is that in these type of tasks here, you would have a deficit if you go in and cause damage to the hippocampus and some other parts of the brain. And here you can see a visual of it, and you can see once again they're just kind of having to remember the significance of these shapes. Which one is going to, you got to remember which one to choose here so that they can actually get the food. All right. And here you can see the performance deficits of monkeys with large bilateral medial temporal lobe in this delayed match to sample. There were significant deficits at all but the shortest research interval. This interval is how long they're allowed to look at and remember the correct choice, but as you make that interval longer and longer to where they actually have to perform, they get worse and worse. At least that's the idea. Let's see here. Okay. And here you can see they're just showing you different parts of the medial temporal lobe. Once again, the temporal lobe, like I said, it wraps around. The amygdala is quite important here and it's interesting. The amygdala, here's the hippocampus, right? The amygdala is right there next to the hippocampus, and remember the amygdala is involved with things like emotional responses and stuff like that. And it makes sense that it would be really close to the hippocampus because. And there, that's shown here. See hippocampus. There's hippocampus right there, right next to the cortex. You probably, in your own experiences, know that when you have a memory that has a lot of emotional content to it, something significant in that sense, that you can remember those things a lot better, or at least you have a vivid account of things than memorizing a list of numbers or something like that, which doesn't provoke an emotional response. So. Yeah, a lot of times we remember things that can be really joyous occasions or really bad. And here you can see they're just showing you more methods here. You got your aspiration, means you're just taking a big chunk of the brain out. That's what you're doing there. They're just showing you more setups. How, once again, you can go through this. It's just how animals are tested. But here you can see medial temporal cortex removal. You get severe deficits in object recognition. But if I just, so this is just getting down into more of the function for each. So the temporal cortex, the cortical part, is more of you get more of a purely cognitive deficit. You might be able to see it and remember that you saw something, but it's hard to remember what it was. That's what they're showing there. Okay, I'm going to just kind of go through some of this here. Yeah, hippocampus, more stuff. Hippocampus also plays a role in spatial memory. Now, spatial memory is just, literally remembering the geography of an area. I always like to say it's where you parked. So you're at Disney World whenever they reopen, and you're out there and they got massive parking lots. Of course, nowadays you're going to take pictures and keep it on your phone so you remember where you parked. But if you didn't have that, it would be harder to find your car, I guess. At least in terms of just remembering the geography. All that kind of stuff. Here you can see. Now here, they show you here storing a food cache. Yeah, the bird has to remember where it was put. So this would rely on the hippocampus. You got to remember where it's put. Let's see here. I want to get through. So some brain areas that are implicated in memory. Inferotemporal cortex, that's basically that's the bottom of the temporal lobe. The amygdala, talked about that, and other parts are here. The prefrontal cortex, that's the very front of your head. The cerebellum is definitely involved with memory for like motor functioning and stuff like that. Like I said, riding a bike. walking, things that appear to be effortless, but you had to learn how to do it. So a lot of motor functions there. And here are some of the different areas of the brain that are involved in different types of memory. You can see there's a lot. There's the cerebellum. Here's that temporal lobe again. And the bottom part is the inferior temporal. Then the medial temporal, medial just means in the middle. There's that amygdala. There's the hippocampus, prefrontal cortex, all this stuff. So there's an awful lot. It's distributed out. I mean, all these things work together. So it's not like, your memory is just right there or something. It's all of these structures. It's a distributed network of structures that are involved with forming memories and stuff. Now let's see here. Yeah, long-term potentiation. So that's giving you some, so you just found out a little bit of the anatomy, what's going on there, different structures that are important. What about what happens to the individual cellular response? That's the physiology. And one of the things we've known for a long time, at least since like the 70s or the 80s, is that the theory goes is that after a learning event like classical conditioning or operant conditioning or what have you, that you have a change in the physiological activity of the cells. Generally, in certain parts of the brain will show this change in activity, and some parts do not. Like, for example, hippocampus will show this. So hippocampal cells, after, at least the theory goes, is after a learning event will show this long-term potentiation. Basically, it means that you have elevated activity that is the result of a learning. This was of a learning event. Okay, and you can see they're showing you here, rat hippocampus right here. There's the hippocampus. And what you can do is, with electrodes, you can induce learning. You can simulate what it's like for these cells to be a learning event by just simply stimulating the certain pathways in here. They don't show it, but it's called the perforant pathway. Well, actually, no, they do. There's the perforant pathway. By stimulating that, you're mimicking what might be occurring in a regular learning event. At least that's the idea. So you can see here, piece of the rat hippocampus. You have these different areas involved. The pyramidal cell, this is part of the hippocampus. Dentate granule cells is another area that's near the hippocampus. And then pyramidal cells is part of the cortex. Basically, during a learning event, you have an enhancement of this circuit that can last a long time. That's the idea. That's why it's called long-term. So it's basically like, and here you can see we can start stimulating the perforant pathway, and after you do this, you get a change, an elevated activity in the cortex and some other parts of the brain there. And the idea there is that, okay, maybe this is the physiological basis for learning events, is this long-term potentiation. At least that's the idea. Some receptors that are important. Here you can see the NMDA, definitely important. NMDA receptors play a critical role in learning, memory consolidation, and things like that. Just as a side note, alcohol tends to interfere with NMDA functioning, which explains why people can black out if they have an awful lot to drink, right? Because you're interfering with these NMDA receptors to function normally, and that tends to cause memory loss. All right, so just to let you know, here you can see NMDA receptor mediated LTP. So the idea is, you have this long-term potentiation. And we say that the important receptors in these neurons, like let's say it could be in the hippocampus or whatever, the NMDA receptors are activated. And here you can see, here's release of glutamate and is binding to the NMDA receptor. And then that could open up what's called, you can see it's a calcium channel. So calcium goes in, and it usually causes long-term changes in the cell. All right, and here you can see. And so these NMDA receptors are quite important. They're activated. Calcium can go in there and initiate long-term changes in the cells. And you're getting changes presynaptically from the sending cell and postsynaptically from the receiving cell. You also find with the learning events, anatomical changes. You have the synaptic connectivity tends to get more complex. You get more spines, more dendritic spines. You get an increase in the number of postsynaptic receptors. This all has the effect on making the connections there much more elaborate and sophisticated. Incidentally, one theory is that during, let's say, depression, let's say if someone is depressed, I know that one of the problems with depression is you can have cognitive problems. You can have problems with, learning things and remembering things. That's why some folks that get depressed, that have a clinical depression, might have a hard time at work or a hard time thinking. Maybe they would have a hard time in school when they got to think a little bit. So you have the opposite. You'd have a decrease in the number and size of the synapse, a decrease in spines, a decrease in postsynaptic receptors. That's what we find. At least that's the theory for depression. Here it is, right? So here we see prolonged low-frequency stimulation of presynaptic neurons. That can actually induce less activity. So depending on how you stimulate the cells, you can get either an increase in activity over time or a decrease. Okay? At least that's how it goes. Okay, let's see here. Once again, if you have any questions, I know I'm just sort of throwing this at you. If you have any questions, please email me or whatnot. This is interesting. You might, if you get some tea, it might have ginkgo biloba in it or ginseng. These are, these are considered to be natural ways to help memory out. It's not reliable. I actually had a professor when I was at University of South Florida, a guy in the biology department who was kind of obsessed with all this. And he had me pull a bunch of papers about, ginkgo biloba and ginseng. And it's really there's really not much there in terms of an actual effect. But maybe it works, I don't know. Okay, so I just went through that pretty quick. If you have, like I said, it was just a quick overview. And you can see just real quick once again, make sure you study all this stuff. If you have any questions, please email me. Keep working on those term papers. Keep doing that, and just take care. And I hope you're all doing great. And I will load up a couple of more lectures here, and I hope these help. At least give you an idea of some of the major concepts here. I guess before I stop it, just remember that the brain is not. It does not work like a computer. It adapts. It changes constantly. These receptors change. The number and how they work can change, and all this stuff can change. So the brain really is quite a marvelous device. Anyhow, that's it, and have a good day.